Good afternoon, everybody. I am going to be going over part of chapter 1 for our course. This is the first section going over A&P Basics for this class, and noting that you have assignments due this week that relate to these chapters. This is going to be smart books, video reflection and responses, and a discussion post. So we'll start with chapter 1. So let's look at some of the origins of where anatomy and physiology may have come from before we get into some definitions. So a lot of our early healers, these would be our doctors, nurses, anyone in a medical profession considered like a clinician, they depended mostly on superstition and magic as to where they think certain diseases or how the body worked. And so we can even see kind of an example here, right? This is during plague times. People thought that fire, the production of smoke, cleared the air to prevent the spread of plague. We now know with modern medicine that this is not true, okay? That this smoke and fire may have scared away rats that carried fleas that carry the pathogen that causes the plague. And that's why it might have seemed effective, even though it wasn't truly clearing or cleansing the air, but it may have been driving away the populations that contain these vectors. So, again, a lot of early medicine didn't really know why things occurred, okay? And as we continue in this profession, these are followed by observations of injuries, wound healing, and cadavers. These are dead bodies, right, that people would use as anecdotal evidence to try to figure out what is going on with the body, what is causing certain diseases, what's going on. And so this evolved into different experimentation, a lot of which involved cadavers and corpses, right, in order to get knowledge about the human body. And we developed terminology for anatomy and physiology as the study we know it today. And even, in early studies, right, 1543, all right, Vesalius actually drew this illustration, right, of different muscles in the human body. And even though this was a long time ago and they didn't have the same equipment or advances in technology that we have now, this is very accurate, okay, for different muscles, major muscle types that we see in the body that you will learn eventually in this class. So, a lot of what we know now from anatomy and physiology came early on from the study of cadavers and corpses. So let's get into some of these definitions, okay? So what we came up with for anatomy and physiology and why we might study it, and this is to help with health outcomes, to understand the human body, those types of things. We have anatomy, which is the study of structures. We would call this the study of a structure or morphology. That would be the structure or shape of something, of the human body and its parts. And this word anatomy is derived from the Greek for meaning a cutting up, so a piece of something. And so whenever we refer to anatomy, I'm discussing anatomy concepts. This is going to be the physical body parts that we're talking about. And you'll notice in the lab with our lab list, we are focusing heavily on anatomy, which is just memorization of the parts of the body. Physiology is a little bit different. Physiology requires a little more critical thinking as to why these parts work together and for what function, right? So physiology is actually the study of the functions of the human body and its parts. And this word is also derived from the Greek, meaning its relationship to nature. So we might know the anatomy of a body part, the shape of it, what it looks like, but we also want to understand its physiology. Why is it shaped that way? How does it function to keep the human body working? And oftentimes, when we look at our organs, the shape that they are, their anatomy, why they're even named, will tell us what their function is. So we have some examples of this. So here we have your hand, right? And we have a variety of muscles, bones, and joints that are participating in this, okay? Grasping around a bar, okay? And we can see something us humans have, which is this opposable thumb, being allowing us to do this kind of saddle motion, this grasping motion. And so our hands were specifically made in this shape and our joints in these directions to do that grasping function. That's its physiology, is to grasp the anatomy, how our parts are shaped, allow us to do the grasping. We've got another one here. This is looking at the external ear canal, okay? our ears, even though we just kind of see the outside, they essentially create funnels, right? It's wide on the outside and it gets narrower as we work our way into the ear. And this provides kind of a funneling system for sound waves so that they get more concentrated at a certain point, which vibrates different structures and bones in your inner and middle ears, right? In order for us to convey sound. And so the shape, the anatomy of your ear also will help us understand the physiology, how we hear things, how sound waves get processed through the ear. So oftentimes anatomy and physiology are linked to each other. I would say though, for this class, right, you might be taking Bio 117 and 118 at the same time. 117, we focus a lot, not just on the anatomy, but the physiology, the why, the functions. And then in lab, we have a lot of focus on just the anatomy. So just learning the parts, and you learn the functions for those parts in lecture. So let's talk about another concept that's pretty strong in anatomy and physiology, and this is the idea of homeostasis. And I give you a quick definition here. Homeostasis is the maintenance of a stable internal environment. So this is referring to homeostasis internally, inside the body, okay? And how our body handles either external things happening to it or even internal things, right? Say we eat something very sugary and our blood pressure goes up. Our blood pressure doesn't stay up forever. Our body processes that sugar, produces different hormones to take care of it, and in order to maintain and bring us back down to normal blood sugar. And so this is what homeostasis is talking about, maintaining stable internal environment. We could have some external things, right? We could get really cold, right? We might step outside, it's freezing outside, it might make you shiver, right? That is a homeostatic response, right? Your body is trying to maintain homeostasis, stable environment. And so it's going to trigger different things, like maybe triggering your muscles to shiver to try to produce heat. So that's what we're looking at here. And we have with homeostasis, okay, our basic definition, but we also have what we call homeostatic mechanisms. These are self-regulating systems that monitor the internal environment and correct as needed, right? And for homeostatic mechanisms, we have two names for them, which we'll get to on the other slides. The first one is negative feedback, and the second one is positive feedback. So keep that in mind. Those are our two examples for homeostatic mechanisms. Before we get into each of those examples, let's just break down what a homeostatic mechanism is doing. There are three main parts. First, you need a part that's actually going to detect a change of some kind. We would call this a receptor in the body. We have lots of receptors inside the body, but if we think about our temperature example, say it being cold, we have receptors in the skin that feel changes in temperature, okay? And so, excuse me, this receptor detects and provides information about the stimuli. The stimuli is anything that's changed, okay, that causes the receptor to react. So maybe we're cold, right? Okay. We have a receptor that detects that, right? And the stimulus is the change in temperature. This receptor is going to send signals through the body, and we're going to talk about our organ systems a little bit later in this chapter, but it'll send signals through the body to your control center, which is always going to be your brain and spinal cord. Okay, but we'll get to that. Your control center is going to process the information the receptor is giving it. It's the decision maker, and it will tell us what to do to bring us back down to the set point, to our stable internal environment. And it does this by communicating with our last part of the homeostatic mechanism, which is an effector. Effector is a muscle or a gland that's going to respond to the decision that was made by the control center and make a change so that we can go back to our stable internal environment. There's a really good example here, also relating to the skin, but we might think about pain, okay? Say I step on a nail. I have pain receptors in my foot that are going to detect and provide information about the stimuli. So I stepped on a nail, my pain receptors felt that, I recognize that it's a nail, and they send all this information to the control center, my brain and spinal cord. The control center is going to say, we don't want to stay stepping on this nail. This is painful, right? And it tells us what to do. It communicates with an effector, which would be the muscles in my leg and foot, to move away from the painful stimulus, the nail, right? To make that necessary change so that I'm not in pain anymore, okay? And all of this, me receiving the signal with my receptor, sending it to my brain and spinal cord, and then sending a signal to a muscle, that could all happen in a millisecond, right? I don't step on a nail and have to think about it for a few minutes before I finally move my foot away, right? This is a very quick process in order to maintain my body's stability. So we have this in kind of a picture format, if that's a little bit easier, right? We have some sort of change that occurs, which we would call a stimulus. And you saw in the previous slide, it said stimuli. That would just be multiple changes, plural. The receptors detect it, all different kinds of receptors for different changes, sends that signal to the control center. The control center sets the set point. Your body temperature is, roughly, right, 98.7 degrees, something of that flavor, roughly. Okay, we might have a little bit of deviation from that. That's our body's set point. So if our body gets a fever, we get sick and it goes up higher, or if we get really cold and it goes lower, your body's going to recognize that. It has different temperature receptors, and we're going to try to go back to our set point where we want to be. And it does that by using effectors, muscles, glands, other things that are going to respond so that we can make sure that the change is corrected. So let's look at our two mechanisms. So we have two homeostatic mechanisms, negative feedback, which is the most common, and then we have positive feedback, which is only in special circumstances. So for negative feedback, okay, this is the most common type. This is what your body does all the time in order to go back to its normal range, okay? So we have those effectors, right, that will return conditions to the normal range, and the deviation from the set point lessens. We can kind of we call it negative feedback because the response moves in the opposite direction. So say I were to draw here briefly, okay? So say we have a change which causes us to increase our temperature, okay? Negative feedback says that we want to do a change, our effectors set a change that goes in the opposite direction because we're going to try to go back down to normal temperature. And so that change will look like this. Another arrow going in the opposite direction, right? So that we go back down to our normal temperature. It could be the same way, right? Say our temperature goes too low. Your body's going to try to do some changes using effectors to go back up so that we reach that set point again. So this is kind of what negative feedback looks like with these opposite moving arrows, right? Say we take for this example here with temperature. Say you go outside and you don't have a coat and you get really, really cold. Your body's response, okay, because primarily most of your heat is generated by your muscles, okay? So your body's response when you get very cold is to send a signal to effectors, these are muscles, to start shivering to increase the heat production in your body and try to bring your temperature back up to normal, okay? This is going to help us using negative feedback, which our body does all the time to maintain homeostasis, prevent sudden severe changes in the body, right? Because we're going to just try to go back to where we normally are. And really good examples of this, right, is body temperature. Your blood pressure is another one. Your glucose, your blood sugar is another good one, where if we go too high or too low, we always try to go back to that even area. This is negative feedback. Positive feedback is going to look a little bit different, okay? And hopefully, yep, we might have to erase this. That's fine. And so if we want to go ahead and look at positive feedback, which is our other one, and remember, positive feedback is not very common, okay? Positive feedback is going to be something that occurs during special circumstances in the body. And it's not necessarily going to bring you back to homeostasis, even though it is one of our homeostatic mechanisms. And what it tells us is that the change, right, that we're seeing in our body is intensified instead of reversed, right? We saw with positive feedback, something goes too high, we do our other arrow down to bring it back, or vice versa. Positive feedback does not look like this, okay? It looks kind of like this. We have something in the body going really, really high, and we intensify our response by doing even more. Okay, so these are both going up for positive feedback. There are very special examples for this. Our body does not do this all the time. And the effect for positive feedback is generally short-lived because we don't want them to stay around forever. So we can kind of see it's an intensified response, but for a short period of time, and then we go back down to normal. This will produce unstable conditions. Okay, so not homeostasis, but eventually we will go back down to homeostasis. Some examples we have of this is blood clotting and uterine contractions in childbirth. We're going to talk about blood clotting a bit later in this class, but let's use the example of uterine contractions during childbirth. So we'll go to the next slide. Oh, let's see. I may have to draw this. Apologies. So in your book, there is a great photo, okay, of uterine contractions for childbirth for a positive feedback. So some of you may not be familiar with childbirth in this class, okay? But uterine contractions are controlled by hormones, and a really big one is oxytocin, okay? You may have heard of this. You may have had a child yourself, right? Some of our females in the room. And sorry for my handwriting, but we've got oxytocin, okay? This is a hormone that is going to be in high levels during childbirth. And for uterine contractions, usually they're stimulated, okay, by the baby's head pushing on certain body parts, okay? So as the baby is making its way through the birth canal, it is pushing its head and stretching different parts. And those are detected by receptors, okay, stretch receptors, and they are going to trigger the release of oxytocin. And instead of us going back down, okay, right? We have such high levels of oxytocin. Let's try to bring them back down and go back to normal. We don't want to do that in positive feedback because we want to accomplish something that's short-lived, which is childbirth, okay? We hope it's short-lived, right? But we're trying to accomplish something, and when it's accomplished, we can go back down with our levels. So for oxytocin, the more the baby's head pushes on parts of the birth canal, the more oxytocin is produced, okay? This is an intensifying reaction because we want to reach an outcome of where we give birth, okay? That would be the outcome of this. And once you do give birth, then these levels can go back down to normal and we can go back into homeostasis. So this is just an example of positive feedback only happens in certain cases, okay? But what is the more normal one that we're going to look at, right, is going to be negative feedback on that previous slide. Negative feedback is what our body does every single day, right, in order to maintain homeostasis. Positive feedback only in special circumstances. And again, we will get into blood clotting a little bit later, okay, in some of our later chapters for this. This is going to be it, okay, for our first portion of chapter 1. Next week, we'll be going over regional anatomy and directional terms, okay, which will be a little bit more content. But for this week, all I want you to kind of focus on is the basics for A&P and homeostasis, which keeps our body going. Thanks.